After we map the RNA-seq into the genome, quality control the RNA-seq sample, and quantify the transcript abundance, we can then evaluate differential gene expression. In order to do this accurately, we need to model the RNA-seq read distribution across the genome. In early expression microarray days, data was not modeled as normal distribution. This is quite different with RNA-seq reads, where from the previous sections, you can see that if we use this Kij value to measure the read count or the fragment counts that's aligned to gene I in sample J, we know that this value is proportional to the expression level of this particular gene, to the length of a gene, to the sequencing depth, which is the library size. There are other factors that are influencing this value. This includes library preparation factors, for example, PCR amplification, some in silico values such as, or factors such as the sequence alignment algorithm. And one additional factor that's important is the library composition, which means that usually if you have one gene that is expressed at very, very high level, it will suck the reads out of other reads because your total library size might be set at certain number of reads total. And therefore, the expression level of one gene might have some indirect effect on other genes because they compete for the total read to be sequenced. As we mentioned in the early days of microarray, people modeled the expression index distribution. For that, they could look at the expression of all the genes in one sample and plot their expression value. They could also look at the expression of one gene across hundreds of other samples to see what is the distribution. And depending on the algorithm for the classical method for microarray expression index, it was determined that the expression data for microarray is normal distribution. With RNA-seq reads, if you take the log value of the count on the gene, it does follow a normal distribution. So which is the case, a lot of data after you take log, you see normal distribution, but maybe this is kind of transforming the data too much that you might lose some power to detect differential gene. And so people want to understand the raw data distribution. And so before we do that, we want to show people how to really test whether your data follow a normal distribution. This is by a QQ plot. And so on the x-axis, we can plot the theoretical quantile of in this case, expression data, which is what percentage of genes have, or what percentage of samples have an expression level below a certain value. And so in this QQ plot, you can imagine you expect more things to be close to the center, right? Like this in the histogram, and then fewer samples nearby. And so you check every quantile to see, do I have the same percentage of samples below or above a certain level? But if the raw data look like something like this, you can see the curve is mostly above the diagonal, but if it's very right skewed, you can see that this data is very much below the diagonal. So if the overall distribution is of the theoretical normal distribution quantile, this indicates that the data is not normally distributed. In fact, RNA-seq data is not. And so we can think about other distributions to evaluate the data. So we know if you think about a scenario where in emergency room, the likelihood of emergency patient going into the emergency room in a very small interval, like 10 minutes or 20 minutes, it's actually a small probability. Therefore, when you look at a bigger interval, such as an hour or two, you ask, what is the chance or what's the probability I'm gonna see X number of patients arriving at the emergency room? This follows a Poisson distribution. And so people early days believed that with RNA-seq reads, we probably also expect a Poisson distribution because say within say 1 kb region, the likelihood that you will see some reads, it's a small probability, especially in the early days when we were sequencing only maybe 3 to 5 million reads out of a 3 billion base genome. So it's a small probability that you have a read lands in a genomic interval. Therefore, when you have a slightly longer interval to say in a 3 kb transcript, which is the average RNA transcript length, what is the expected number of reads you see on a particular gene kind of follow the same distribution, which is Poisson distribution. So in standard Poisson distribution in here, we only have one parameter, which is the average events per interval. And if you then ask what is the probability that I'm going to see k number of events in this interval, this follows a Poisson distribution, and the variance and the mean all use the same parameter lambda. And so we want to show you an example of this. So the probability of k events in this interval will follow this Poisson distribution formula. And so for example, is if the expected value or the unit interval for this is only 1, you can see here we have about 37% or 6% chance of not seeing a single event, which means, in this 1 hour, no patient show up in emergency room. But also there's about similar probability that you will see one patient. And then as you go further and further, the probability decreases. Whereas if the lambda is equal to 4, we will see this distribution, and then the lambda is 10, we'll see this distribution. You can see compared to a normal distribution that we see in here, the Poisson distribution have a wider spread, and it also is not evenly distributed across the mean. So this is the standard Poisson distribution. Interestingly, for RNA-seq reads, we can first draw a figure like this, where if you look at expression data in many, many different samples, and the x-axis is the average expression of one particular gene across the samples, and we take a log, and the y value is the variance of this gene across all the samples. Say if you measure many, many samples in one big study. And each dot here represents one gene. So you can see here, generally, if a gene is expressed at lower level, it has lower variance. If it's expressed at higher level, it will have higher variance. If this follows a Poisson distribution, we would expect all the genes to line up on this black line, which means the mean and the variance are the same. Instead, in real RNA-seq data, we see something like this, which suggests a over-dispersion, which means that when the expression level is higher, the variance is even bigger than the mean. And a negative binomial distribution can be represented by NB, R, and P. Classically, negative binomial distribution is defined as if the probability of success is a P, the number of successes before the first R number of failures. And so if we want to see, what is the probability that in this, we are going to see a particular K. And so we know that the parameter, the formula for the negative binomial distribution is like this. And it's a little bit complicated compared to the Poisson distribution, but with negative binomial distribution, you can also have a mean and a variance. And in this case, the variance is, when the mean increases, the variance is actually higher than the mean. And this can be demonstrated by this figure in here. You can see when the expression level is low, you can't really go below 0, but here is the distribution. Whereas when the kind of the expected level is higher, the distribution can also have a wider spread compared to, for example, a Poisson distribution. You can see here, when the negative binomial at 10, the spread is much bigger compared to the Poisson distribution at 10. And so using a better distribution to model the expression which better capture the actual gene expression, we will then have better statistical power to detect differential expression between the different conditions. And we will continue that in the next lecture. So today for the lecture, we discussed the RNA-seq experiment and how to quality control your RNA before you put this in the sequencing library. There are some design considerations and also the importance of having biological replicates. We discussed how to do read mapping using a splice-aware read mapper instead of BWA, which map to the reference genome just on the DNA level. And after read mapping, we then do quality control of the read, and this is using RCQC. In order to quantify the transcript, we can use the more accurate but slower algorithm RSAM, or we could use pseudo-aligners, which give you kind of a rough idea, such as Callisto and Salmon, which improve the speed but decrease the accuracy. In terms of expression index modeling, in the early days, people use RPKM or FPKM. But now the standard is to use TPM to represent gene expression. However, to calculate the differential expression, people actually use count matrix directly, the normalized read count. And so in order to really look at the differential expression, we need to understand the read count distribution of RNA-seq, which turns out it follows a negative binomial distribution with overdispersion, meaning that when the mean increases, the variation increases even more. Okay, that's all for today.